He has come to this hall. He is MD, DM. He is actually did his MD from Government Medical College, Trivandrum, DM from KEM Mumbai. He is a consultant endocrinologist, Aster MIMS. He is a very good organizer. He has received many awards and published articles in international and national journals, and he is a good speaker. And he is a recipient of AR State Award 2008. Over to you, Vimal. He will be talking on Demystifying Biosimilar Interchangeable Glargine, an Indian perspective.
Good evening. At the outset, I am extremely grateful to Jodev sir and his team for inviting me to discuss something about demystifying biosimilar interchangeable Glargine. And I think I should start with paying tribute to my teacher, that is Joy Phillips sir, who was a fantastic person as well as a fantastic teacher. And now we will discuss something about demystifying biosimilar interchangeable Glargine, an Indian perspective. So I have been supported by Mankind Pharma for this talk. And we will just discuss the topic in the following headings. What are the barriers to insulin therapy? Basal insulin initiation, what are the advantages? Biosimilar insulins, how do they compare with each other and with the originators, and some practical considerations. We know that Indian real-world data shows that India is far from adequate as far as insulinization is concerned. We know the landmark trial which showed that Indians are not having any good glycemic control. Only around 20% of the Indian diabetics are having good control. So if you are seeing 5 diabetics, 1 out of 5 is only having good control. Rest everybody is uncontrolled. So probably we need not be worried about that, because even if you go to US or somewhere else, the data may not be that great. Though they also may be getting around 50% control only. And average diabetes patients is obese as well as overweight. And around 20% of the people will be having complications, and around 5 times higher risk of complications is there in overweight patients. This is what the landmark trial showed. And we know that for every 1% decrease in HbA1c, there is substantial reduction in the complications, both in the macro as well as in the microvascular complications. And one thing that we don't take into account very seriously is the peripheral vascular disease. So you can see the data there, which shows that peripheral vascular disease is coming down drastically to the tune of around 40% if you decrease the HbA1c by around 1%. So that tells us to focus more on food in the evaluation of diabetes mellitus. And insulinization in India is, again, it is not at all adequate. We can see that only around 30% of the people, diabetic people, are managed with insulin, and people who are requiring insulin also are very reluctant to be put on insulin. So if you are seeing some patients who needs insulin, only around 1 out of 2 will agree for insulin. So that is the status of India. Probably it's a needle phobia that is putting them away from being insulinized. And also it's pretty clear that adding OADs beyond 3 OADs is not going to fetch you any result. So this is probably an interesting data again from landmark trial. If you are having a patient who is on 3 oral anti-diabetic drug, and another patient who is on more than 3 oral anti-diabetic drug, the change in HbA1c is less if the patient is on more oral anti-diabetic drug. So you are seeing here, there is a very interesting data. So if you are on 4 anti-diabetic drug, your HbA1c control is inferior to than being on 3 oral anti-diabetic drug. Probably at this stage of time in which the patient is on 3 oral anti-diabetic drug, there is a time in which you should think of starting insulin for that particular patient. So it's very clear that we need to start insulin pretty early in the natural history of diabetes mellitus, and there are so many barriers of insulinization, healthcare provider specific barriers to insulin initiations, like lack of knowledge. So we are very busy in our OPDs, so we may be seeing so many patients. We may not be able to dedicate sufficient time for explaining the insulinization, the risk of hypoglycemia and the technique and everything. So that is one of the barriers for insulinization, and there can be language barriers and concerns about patient compliance and everything. But the main barrier might be the worry from the physician's perspective as well as from the patient perspective regarding the hypoglycemia risk. That is one of the important barriers for insulinization for patients who are being started on insulin. And also there is some reluctance to initiate insulin therapy from the doctors as well. And what we can do is we can explain that insulin helps to control diabetes mellitus. It is not your personal failure that has been the reason for insulin initiation, and we can demonstrate, either you yourself can demonstrate or you can employ a team. So ideally there should be a team of diabetes educators who can provide good education regarding insulinization techniques, and we should establish trust with the patient, and we should explain that injecting insulin is easy, and being available for support, and there should be a team who will be supporting you for guiding you regarding the dosing as well as how to tackle hypoglycemia and everything. So everything should be placed while we start the patient on insulin. And when to consider insulin type 2 diabetes mellitus patient, I think this slide you might have seen many times. When there is increase in HbA1c above target within 6 months, despite dual or triple oral anti-diabetic drug therapies, don't go for to the fourth drug. Just start the patient on insulin. That will be the best thing. When the patient is having significant sarcopenia or cachexia and complications, you have to start on the patient on insulin. Early manifestation of diabetes mellitus, when the patient starts with very high sugars, like 500 sugars with significant metabolic symptoms, it's better that you put the patient on insulin, then you can even think of switching over to oral anti-diabetic drugs. When there are so many subgroups, I think you might have heard lectures about the sub-classification of diabetes mellitus, and there are some subgroups in which there is significant insulinopenia. Those sort of patients in whom there is a significant insulinopenia, it's time that it is good that you put the patient on insulin. And if there are symptomatic patients also, it's better that you put the patient on insulin. So a patient is coming to you with a blood sugar of more than 500 and patients having significant polyuria, polydipsia and everything. Will you start the patient on oral anti-diabetic drug? Probably no. That patient, the best treatment will be to start the patient on insulin plus or minus oral anti-diabetic drug, stabilize the patient, and then go for whatever as the treatment of choice in the coming days. So what are the 4 crucial factors which significantly influence the selection of appropriate insulin for a patient? One is efficacy, that the drug should be efficacious, it should be safe. Safety means hypoglycemia. Hypoglycemia is the most important safety parameter for any patient on being started on insulin. It should be convenient for the patient. It should not be troublesome, or it should not be cumbersome for the patient. It should be affordable for the patient. So these are the 4 factors, crucial factors that significantly influence the selection of appropriate insulin for a particular patient. And what are the advantages of timely initiation? It helps in overcoming the glucotoxic effect of hyperglycemia. It puts the beta cell to rest, and it improves insulin sensitivity. It may alter the course of the disease progression. There may be some effect of insulin on the natural history of diabetes mellitus. It has got some proposed anti-inflammatory and antioxidant properties, and long-term protection to end organs by metabolic memory. Nowadays we talk too much about the legacy effect as well as metabolic memory effect, in which the patient's early aggressive glycemic control will be having long-standing benefits. So these are the benefits, or these are the advantages of timely initiation of insulin therapy. And this is why we say that we should start insulin at the appropriate time in any patient with type 2 diabetes mellitus. So a basal insulin regimen is the ideal start. So if you travel from US to Canada to UK or to Australia, wherever you go, the ideal first start insulin by whatever guiding authority, be it ADA or be it EASD or be it CDA, Canadian Diabetes Association, wherever you go, it is basal insulin, which is the treatment of choice as the initiator insulin. So, and it is the number 1 initiator insulin for across the globe has been Glargine for last 30 years. And Glargine 100 provides better glycemic reduction versus human premixed insulin. And Glargine 100 provides similar HbA1c reduction when compared to even co-formulation as per some studies. And it is extremely safe. Hypoglycemia is extremely less. And it is convenient because patients usually the basal insulin we take at bedtime, so that the patient has got can do all the work during the daytime and relaxed at home. He's relaxed at home. He has his dinner, and after dinner he takes the insulin. That finishes the job. So it's extremely convenient. Nowadays it is very affordable as well. So that's the point. It's highly affordable as well. Previously it was expensive, but nowadays it's highly affordable. And there are some speculations which said that as far as the HbA1c goes up, the predominant contributions from fasting glucose is the international data. But there was some speculation saying that in India or in Asia it may not be true. But still, even in India, it is said that as the HbA1c goes up, the contribution from fasting glucose is significantly there. So that is what the data shows. So even if the HbA1c is more than 10%, the fasting glucose contribution is around 60%. So definitely, even at higher HbA1cs, we definitely need to tackle the fasting glucose. So fasting glucose tackling is extremely important. That the best treatment option for tackling the fasting glucose is basal insulin therapy. And I think we might have heard many times regarding fixed fasting first. So what is fixed fasting first? So once you tackle the fasting glucose, once you bring down the fasting glucose, the overall 24-hour glycemic control is going to be controlled fairly adequately. So that is the point. So once you control the fasting glucose, even the postprandial peaks will come down and the patients will have a holistic glycemic control for the 24-hour period of time. So holistic glycemic control can be achieved with basal insulin, and this has been there in my practical experience as well. So one of the insulins that I have been trying in my clinical practice over the past 15 years is starting with basal insulin. It has been fetching results for the past 10 or 15 years of my clinical practice, and basal insulin has been always been good for as an initiator insulin. And there is a huge amount of evidence in favor of glargine. I think this standard molecule, I need not explain to this august crowd regarding the benefits of glargine. And if you search PubMed, you can see that there are 925 clinical trials, 822 RCTs, and 135 meta-analysis, and 694 review articles which have been written about glargine. So there is no doubt that it's the best insulin to be released into the market in the past 30 years. And glycemic targets achievement with basal insulin has also been fairly good. So you might think that if the HbA1c is around more than 8.5 or 9, glargine will not work alone, because we know that Indian population have predominant PP sugars. So we might always have the feeling that we are not going to get glycemic target achievement with the use of basal insulin alone, but that is not true. Even if you are having an HbA1c of around 8.5 or 9, you can have an HbA1c reduction to less than 7% with the use of glargine alone with oral antidiabetic drugs. This is evident in many trials like INSIGHT trial, TTT trial, Apollo trial, Laptop trial, triple trial, so many trials which have shown that even if the HbA1c is around 8.5 or 9, you can have a good glycemic control with the starting glargine alone. And now we'll talk regarding what is biosimilar insulin. So we know that glargine or any sort of insulin is a biological product, unlike aspirin or paracetamol, which are chemical products which have got a small chemical structure. The molecular weight will be very less. But here we are having a biological molecule which has got a large structure and which is produced from living organisms and carefully monitored to ensure consistent quality. So that is a biological product. And there are innovator molecules like Lantus is an innovator glargine. And biosimilar insulins are similar to reference product, and they have been characterized by physicochemical characterization. They are similar to reference product. They are not exactly same. And they are approved by FDA after rigorous evaluation and testing and functional characterization and clinical performance. This is how a biosimilar insulin is developed. And what are the physician's perspective to consider a biosimilar insulin? First of all, it is the indication. For example, if a person is having a fasting glucose of around 100 with a postprandial sugars of around 300, will you start the patient on basal insulin? Probably no. The patient who is having a high fasting glucose with the corresponding PP sugar elevation, then only we will consider basal insulin as the initial insulin of choice. So definitely indication should be there, and the patient should be having a sufficiently high fasting glucose to start the patient on basal insulin therapy. And the biosimilar insulin should have the proof of safety should be there. They should have undergone rigorous clinical trials to show that they are safe, and they should have evidence of bioequivalence with innovator molecule, and they should have proof of efficacy by comparative studies with the innovator molecule, and they should also undergo immunogenicity testing. So what is immunogenicity testing? Any comments? So immunogenicity means sometimes insulin molecules can produce antibodies. So that is immunogenicity. So we should prove that this sort of molecule, the biosimilar molecules, are not producing any anti-insulin antibodies when it is injected to patients. So immunogenicity study should be there in place to show that there is no immunogenicity with the molecule. So with all these criteria should be satisfied, then only we can start the patient confidently on a basal insulin analog or a biosimilar insulin. And even we might think that biosimilars don't have a regulatory requirement, but it's not so. Original development definitely demonstrates safety and effectiveness across so many trials. But even biochemical biosimilars, there are in vitro studies, we call it as test tube studies, then in vivo studies, clinical pharmacology studies, and clinical studies should be in place before getting the approval of a biosimilar molecule. And factors for development of biosimilar insulins in India. In India, definitely there is a huge scope for biosimilar insulin because the basic reason is the population in India. We know that there is a huge diabetic population that is happening in India, and there is a large patient pool. There is a high incidence of diabetes mellitus to the tune of around 10 or even up to 15% in a study done in Kochi. And a robust regulatory environment is there, and state of art in technology is there. India is advancing in technology as well, and there is a huge access to cold chain logistics is also there. So definitely there is a scope for biosimilar insulin in India, and India will need biosimilars, and there is a increasing evidence for biosimilar interchangeable glargines in India. And we know, I think this point I have already covered. There is a huge wall of evidence for the originator glargine or innovator glargine, and HbA1c reductions from baseline to end of study with innovator glargine has been too huge. You can see that some of the studies are showing even up to 2% HbA1c reduction with using glargine alone. So there is a huge database for innovator glargine. So definitely, if you prove that this glargine is similar, the biosimilar glargine is similar to innovator glargine, definitely it is also going to have the same HbA1c control and safety parameters. So what is interchangeability? So now I will tell what is interchangeability. Interchangeability study shows how patients do when they are switched back and forth between the reference product and interchangeable biosimilar candidate. So here we are doing studies with the innovator molecule. In between, innovator molecule will be broken, and instead of that we give reference. So initially biosimilar molecule will be given to the patient. In between the study will be, the patients will be given reference molecule. Then again the patient will be given biosimilar molecule. This is how the study is designed. One of the studies of interchangeable study is known as INSTRIDE-3 study. And it will again detect the development of anti-drug antibodies as well. And also it will also tell whether there is a decrease in the efficacy of using the biosimilar insulin. So this is the interchangeability study. And regulatory approval pathway of interchangeable biosimilar insulin is also a bit lengthier than that of a biosimilar insulin. So here interchangeable means you can change from the reference product or innovator molecule to this molecule as per the studies. So here for biosimilar interchangeable product, there is one more step for acquiring approval, that is the switch study. Switch study means you have to try with the same molecule, the reference product as well as in the biosimilar molecule in the same patient. So that is the switch study. So switch study should be in place to say that this biosimilar is interchangeable with the originator molecule or with the innovator molecule. So this is the INSTRIDE studies. The INSTRIDE-3 study is an interchangeable study which is done with the biosimilar insulin, and it was shown that they are equally efficacious without any production of antibodies, and similarly efficacious. There is no increasing incidence of hypoglycemia. There is no increase in any other adverse events with the use of interchangeable biosimilar glargine. So INSTRIDE-1 as well as INSTRIDE-2 are comparative studies between the glargine as well as biosimilar glargine. And now we are seeing a patient, a 48-year-old Mr. Sanjay, a diabetic for the past 7 years with a fasting glucose of 188 and PP glucose of 224. What's the insulin of your choice? Any comments? Fasting is significantly high and PP is not very high. So probably this patient will definitely benefit with the use of a basal insulin. So he is currently on metformin 2 gram per day, Limpride 4 milligram, sitagliptin 100 milligram per day, and should he be put on a fourth oral anti-diabetic drug? Definitely no. As per the evidence, you are supposed to start the patient on insulin. Which insulin for this particular patient? It's very clear that this patient is likely to benefit with the use of basal insulin, and definitely HbA1c reduction is going to be definitely good for this particular patient, because laptop study has shown that there is 25% reduction in the HbA1c with the use of this molecule, even when compared to premixed insulin. And even lesser risk of hypoglycemia. At this point, I think all of us know, because of the NPH component of premixed insulin, hypoglycemia is definitely high. But for glargine, it is definitely less. And basal insulin is associated with a greater risk reduction of diabetes-related complications as well. And why we should think of switching to interchangeable biosimilar glargine? Because of the accessibility, because innovator might be concentrated on the cities, but the interchangeable biosimilar glargines might be penetrated in the even rural areas, and the cold chain system will be properly maintained for the even the biosimilar glargine or biosimilar interchangeable glargines. You can see that it is available across all the countries, all the states of the country, and even there have been traditional refrigeration method or traditional storage methods for insulin have been tried with for the biosimilar insulin, like earthen jar filled with soil and earthen jar filled with water and everything. It's working well. I think you can see the graph on the right hand side in which the traditional methods have been tried, and it is working fairly well for the storage of insulin when compared to the conventional keeping in the refrigerator and everything. So these are some sort of innovative studies with the interchangeable glargine, and even transportation of insulin from pharmacy to patient's home is pretty easier with the introduction of newer methods of biosimilar glargines. And there are improvised blocks for transportation of insulin from pharmacy to home is there. So, ladies and gentlemen, I would like to conclude by saying that interchangeable biosimilar insulin glargine is an additional clinical evidence for evaluation of safety in switching from reference product. So, you can safely use this molecule because the interchangeability has also been extensively studied, and a safe option for switch from reference insulin glargine product, and an effective solution for affordable and accessible insulin in India. Before I wind up, I should tell two more things. I think I forgot to extend my gratitude to my dear Chandini madam for her kind words of introduction. So that's one thing. And second thing is I invite you all for our endocrinology annual meeting in Calicut on October 6th at Hotel Rabis Kadavu. With that, I would like to wind up. Thank you so much. Thank you.
Thank you, Vimal, for this interesting presentation on biosimilar insulins, especially glargine. And we know that insulin resistance, physiological and psychological, as well as the physician inertia, there is a delay in initiation of insulin, and Vimal has actually highlighted that point. But as a clinician, we always think whenever there is biosimilars in market, we will get it at a lesser cost. So that may be, as you have put the last sentence in your conclusion, if there is a cost reduction as well, along with more physiological to our environment and storage, it will be a good option, because India has a large number of patients and we need more insulin and more persons to treat diabetes. Any quick comments or questions?
They are actually, sir, it is not like a chemical product. Chemical product, it is very easy to replicate the molecule, but here it is the process is the product. The process of manufacturing a biosimilar is not similar to that of a reference or innovator product. Because we are using the bacteria or fungi as being used, and the process of production it's a huge process of production of a large molecule of a insulin-like molecule, not like aspirin or paracetamol where it's a small structure. Here it is said that the process is the product. That's what they tell. The process is also extremely important. So that's why it is being rigorously tested for bioequivalence. So that should be done. Now, I think, sir, nowadays the cost benefit is not that huge, because I think glargine has come to price control, but still there is a marginal price benefit is there. That is what I assume. Exactly, I don't know, but still marginal price benefit is there. But I think glargine has come to product price control also the recent past, around 1 year back.
And they also go through stringent guidelines for getting
so I think the point is that even in US, United States of America, they are using this interchangeable glargine in place of Lantus. So even it is approved in even United States there. So the thing is that if you prescribe glargine, previously if you with the prescription of glargine, they will get only Lantus in US. But the same with the same prescription, because this interchangeability is approved, they can get the biosimilars even in US. So that is the difference that has happened over the past few years. So in US, the pharmacists have got a role for deciding which insulin is to be given. So they can give this as a substitution. So this is a change that has happened.